Hello dear students of class 12 science, welcome to biology class. Today we will begin with a new chapter, that is biotechnology and its application, chapter number 12. Clear? So in the previous classes we have learned about the biotechnology and its processes. We have learned about the construction of recombinant DNA, and we also learned about the processes of recombinant DNA technology. So today in this new chapter we will begin with the application of biotechnology. So the modern biotechnology, it's mainly used in the production of different types of medicines, different types of vaccines. That is mainly in the pharmaceutical industries. They are mainly used for the production of different types of medicines, different types of vaccines, for therapeutic and diagnostic purposes. And it's also used for the production of genetically modified crops, for the production of processed food, for energy productions, for waste treatment also. So we have got different application of biotechnology in different field of science. Clear? So the main objective of the biotechnological research are to provide the best catalyst in the form of genetically modified organisms. Means those organisms whose genes has been altered or has been modified. Usually over here we use the microbe to get the measurable product. Number 2 objective is to create the optimal condition through genetic engineering for the catalyst to act, and the third one is to develop the downstream processing technologies to purify the enzymes or the proteins or the organic compound, because whatever the products we used to obtain by the help of recombinant DNA technology, those products has to be extracted, has to be purified, and needs to be analyzed, and finally after that they has to undergo clinical trial or the downstream processing. Only after the clinical trial they can be sent for marketing purposes for human use. So we have got different uses of biotechnology, and just now we have learned about the objectives of biotechnology. So today this class we learn about the application of biotechnology in healthcare or in the field of medicine. So we are going to learn about the application of biotechnology in the field of medicine or in the field of healthcare, and here we are going to learn about the production of genetically engineered human insulin. We are going to learn about the role of gene therapy for ADA disease, which causes SCID. We will talk it afterwards. Then we also learned about the molecular diagnosis by polymerase chain reaction, that is PCR, and enzyme-linked immunosorbent assay, that is ELISA. So first of all we learn about the genetically engineered human insulin. It's in the page number 614. So all of are aware that the insulin is mainly the hormone which is used for the treatment of diabetes mellitus. Clear? So insulin is mainly the hormone which is secreted by the isolate of Langerhans of the pancreas, which is mainly used to control or for the treatment of diabetes mellitus. So first of all we'll just learn about the structure of the human insulin. So human insulin is mainly a protein which is formed of 51 amino acids, because proteins are mainly long chain of amino acids. So it has got 51 amino acids which are organized into 2 polypeptide chains. So those 2 polypeptide chains are polypeptide A and polypeptide B. That means human insulin has got 2 polypeptide chains. A fully functional human insulin has got 2 polypeptide chain, polypeptide A chain A and the polypeptide chain B. And these chains are synthesized together as a single polynucleotide chain, which is called the proinsulin. Not only that, we have got the polypeptide chain A and B, but we have got the extra chain also, which is called the C peptide. That's why it's also called proinsulin. So we have got chain A, chain B, and we have got extra chain which is called the C peptide. So that's why this proinsulin has to undergo processing before it becomes the fully mature functional insulin hormone. Clear? So one more time saying that human insulin has got two polypeptide chain A and B, but it also has got an extra chain which is called the C peptide. So that's why this proinsulin has to undergo processing to become the fully mature functional insulin hormone. So during maturation, during processing, that extra stress of amino acid, that is C peptide, will be removed. Clear? So the extra C peptide chains will be removed, and the two strand, that is polypeptide chain A and the polypeptide chain B, will be joined together by the help of disulfide bond, or by the help of the sulfide bond, which to form the functional insulin. So in case human insulin, we have got the extra C peptide chain which needs to be removed for the insulin to become fully functional. Clear? Okay, now let's go back to the production of genetically engineered human insulin. So I have told you that the insulin is mainly used for the treatment of diabetes mellitus. So earlier, this insulin was extracted from the pancreas of slaughtered pigs and cattle, but those insulin used to produce some allergic reactions in human beings who are suffering from diabetes. Clear? So they have got certain drawbacks, there are certain side effects. So that's why the first human insulin was produced by the help of recombinant DNA technology, we can say. So the first human insulin was obtained from genetically engineered E. coli, Escherichia coli, in 1983 by the American company Eli company. So kindly remember that the first genetically engineered human insulin was extracted from the E. coli cells by an American company in 1983 by Eli company. Okay, and the name of that insulin was known as Humulin, Humulin. So kindly remember the name of the insulin which was obtained genetically. It was known as Humulin. Okay, now DNA sequences for the chain A and B were introduced in the plasmid of E. coli. Plasmid means a vector. Okay, so those DNA sequences which is to form the polypeptide chain A and the polypeptide chain B, they were inserted into the plasmid of E. coli cell, and those DNA sequences which were introduced into the plasmid of E. coli cell, they used to form the separate chain A and the separate chain B. Clear? So DNA sequences for the chain A will form the polypeptide chain A, and the DNA sequences for the polypeptide chain B will used to form the polypeptide chain B. So chain A and chain B will be formed separately, and afterwards after the formation of polypeptide chain A and the polypeptide chain B, they were joined together by the help of disulfide bond. Clear? So these chains were joined through the disulfide bond to form the human insulin and removing a C peptide segment. So by this way, the first human insulin, the Humulin, was obtained by the help of genetic engineering process by an American company, Eli company. So let's go to the diagram. You can see over here, you can see here the extra stress of C peptide chain. Okay, so we have got the A chain, we have got the B chain, okay, and we have got this extra C peptide chain which needs to be processed during the formation of a fully mature functional insulin. So here we have got the B cell pattern molecules. We have got the separate A chain and the B chains which are being joined by the help of the disulfide bond. Now this will become the functional insulin and the C peptide will be released. Okay, now there will be formation of mature insulin, which will be a functional one. Now the question comes over here, what is the difference between the insulin which is being produced by the Eli company and the insulin which is produced within the body? Clear? These questions become examinations, that what is the difference between the insulin which is produced by the American company, Eli company, and the insulin which is produced within the body. So generally I have told you that within the body we have got the polypeptide chain A and the polypeptide chain B, Along with that, we have got an extra stretch of chain which is called the C peptide within the body. So those C peptide needs to be removed during the processing for the insulin to become fully functional one. But those insulin which are being produced by the Eli company by the help of genetic engineering technique, or we can say the humulin which is produced, it only contains the A chain and B chain. There is no need for the formation of extra C peptide or the C chain. Only A chain and B chain will be there, which will be joined by the help of disulfide bond. Clear? So this is the first role of biotechnology in the field of medicine, which is mainly used in the formation of the insulin, which is used for the treatment of diabetes mellitus. Now let's go to the next one, gene therapy. Okay, role of biotechnology in the field of medicines. We'll talk about gene therapy. So gene therapy involves the treatment of a hereditary disorder caused by a single defective gene. So gene therapy is mainly used in the treatment of hereditary diseases, those diseases which are being transmitted from the parents to the offspring, which is mainly caused by a defect in a single gene. So those defective genes can become functional by the help of gene therapy process. Now it involves replacing the defective gene or the harmful gene or the lethal gene by the normal one. The normal gene delivered into the embryos or the person tissues takes over the normal function over and above the defective gene. So those normal genes which we have introduced by the help of gene therapy technique will take over the defective genes, so that the gene will become or will function normally. So gene therapy is mainly of two types. First one is called the germline therapy, and the second one is called the somatic gene therapy. So what is germline therapy? So in germline therapy, functional gene is introduced into the germ cells. Germ cells means reproductive cells such as sperm, ovum, clear, or even we can place the functional gene in the zygote for the replacement of a defective gene, the gene which is not functioning normally. This therapy is carried out in case of farm animals and in laboratory experiments, but not in human beings so far. Number two is somatic gene therapy. In this case, in this therapy, the functional gene is introduced into the somatic cells. Somatic cell means the body cells. Okay, so we have got two types of gene therapy. Germline therapy, in which the functional gene is introduced into the reproductive cells or the germ cells like sperm or the ovum or even in the zygote to replace the defective genes. And in somatic gene therapy, we are introducing the functional gene into the somatic cells or the body cells. Clear? So now we will learn about the gene therapy for ADA disease. ADA means adenosine deaminase deficiency disease. Okay, so the first successful attempt of gene therapy was carried out in 1990 in a 4-year-old girl suffering from adenosine deaminase deficiency disease, ADA disease, and the deficiency of this ADA causes a disease called SCID. SCID means severe combined immune deficiency disease. So kindly remember the abbreviation of SCID, which means severe combined immune deficiency disease. And this SCID is a rare genetic disorder of the immune system. It's also an autosomal recessive disease, which is mainly caused due to the deletion or the loss or mutation in a normal ADA gene for the enzyme adenosine deaminase. That means the gene which is responsible for the formation of the enzyme adenosine deaminase will be non-functional. That means that genes will be lost, or it will be deleted, or it will undergo mutation, so that it will not be able to produce the enzyme adenosine deaminase. Clear? So this adenosine deaminase is the enzyme which is essential for the proper functioning of the immune system, and in the absence of this enzyme, the immune system will not be able to function properly. And why the deficiency of this enzyme? Simply because the gene, that's ADA gene, which is responsible for the formation of the enzyme, is either mutated or is being lost or is being deleted. Okay, so the normal enzyme is not being produced and the immune system will not function in a properly manner. So the disease can be fatal if it is not treated regularly. And the treatment for ADA disease can be carried out by transplantation of bone marrow with normal ADA gene, or enzyme replacement therapy, where we will inject the functional ADA enzyme, or we have to go for gene therapy by introducing the functional ADA gene into the lymphocytes. So here we will talk about the gene therapy process for the treatment of ADA disease. Clear? So in ADA gene therapy, what we are doing is that the lymphocytes, okay, the lymphocytes, the WBC from the patient's blood are grown in a culture outside the body. That means from the patient's body we will take off the lymphocytes and we will culture that lymphocytes, not inside the body of the patient but outside the body. Okay, then a functional ADA complementary DNA, that cDNA means complementary DNA, is introduced into the lymphocytes. One more time, a functional ADA cDNA means complementary DNA is introduced into the lymphocytes using the vector. Now, which vector we are going to use? We are going to use the retrovirus vector for the introduction of ADA cDNA into the lymphocytes. Clear? Now, these genetically engineered lymphocytes are returned back into the patient's blood, and the patient needs periodic infusion of such genetically engineered lymphocyte because lymphocytes are not immortal. So what we are doing in gene therapy for the ADA treatment, we are taking out the lymphocytes from the patient's blood and we are culturing it outside the body. Then we are introducing functional ADA cDNA into the lymphocytes by the help of a vector, that is retrovirus vector. And that genetically engineered lymphocytes are returned back into the patient's blood. But periodically we have to introduce these genetically engineered lymphocytes into the patient blood because lymphocyte is not immortal. Okay, so what is the proper treatment for the ADA disease? If you go for gene therapy, then we have to introduce the genetically engineered lymphocyte at a particular time period because this lymphocyte is not immortal, right? It will not last for through the lifetime. It has got a particular lifespan. During that particular lifespan it will walk, it will be functional. So it is not a long-term effect or the long-term treatment for the ADA disease. So what is the permanent cure for the ADA disease? Because the gene therapy where we are using the functional ADA cDNA or the genetically engineered lymphocyte is not giving the permanent cure or the permanent solutions for the treatment of SCID. Okay, clear? So what can be the permanent cure for the SCID or for the ADA disease? So in that case, what we can do, if the gene isolated from the bone marrow cells of a normal person, one more time I am saying that for the permanent cure of ADA disease, if the genes which are isolated from the bone marrow cells of the normal persons are introduced into the cells at the embryonic stage, one more time I am saying that from the normal persons, if you will take off the gene from the bone marrow cells, and if that gene is introduced into the cells at the early embryonic stage, a permanent cure can be achieved. Otherwise we have to go through the periodic infusion of genetically engineered lymphocytes which is not immortal. Okay, so for the permanent cure of ADA deficiency, we have to isolate the gene from the bone marrow cells of a normal person, and that gene has to be introduced into the cells at the early embryonic stage to get the permanent cure for ADA deficiency disease. Then only it will give permanent cure for the disease. Clear? So here we have finished the role of gene therapy for ADA deficiency disease which causes SCID. Now next one we'll talk about the molecular diagnosis. Okay, so molecular diagnosis will help in the early detection of a disease or the predisposition of a disease. And here we will take the help of polymerase chain reaction and enzyme-linked immunosorbent assay ELISA, which are the technique for the early diagnosis of a disease. It is very important to detect a disease at its earlier stage. Okay. So first of all, we'll talk about PCR. So PCR is mainly a technique where we can amplify or multiply a gene or a DNA. We have learned earlier in the chapter biotechnology processes that whenever we'll try, like we want to amplify a gene of interest, or we want a higher number of DNA having gene of interest, then we have to amplify those gene or DNA by using the technique called PCR. Okay, and over there we have learned about different steps like denaturation, annealing, and extensions. Clear? And we also learned about different type of enzymes which are used in PCR. So try to recall it back. Okay, so PCR is mainly a technique where we can amplify the gene or the DNA for obtaining numerous copies of DNA molecules within a short period of time. Now this technique can be used to detect the traces of DNA and is applied to detect the presence of bacteria or viruses in the initial stage of infection. So at the beginning stage of infections, we can detect the presence of bacteria or viruses in the body by the help of PCR, even when the symptoms of the disease are not visible in individuals. So PCR is mainly used to detect the HIV in suspected AIDS cases, and in case of mutations or changes in the genes or in the DNA in case of the cancer patients. Okay, so here a single stranded DNA or the RNA probe, radioactive probe, is made to hybridize to the complementary DNA in a clone of cells, and the hybridized molecules are then detected by the help of autoradiography. And if there will be any sort of mutation in a DNA, then the DNA cannot form a double-stranded structures. Okay, so here PCR can be mainly used for the detections of HIV at the early stage, or even we can detect the cancers at the early stage by the use of DNA or RNA with radioactive probe. Okay, which is mainly used to hybridize with the complementary DNA. But if there is mutation, I am saying that if there is no mutations, if there is mutations, then it is not possible to form hybridized structures with the complementary DNA, and that will give us the idea whether there is mutation or not. Clear? And let's go to ELISA, the enzyme-linked immunosorbent assay, which is mainly based on the principles of antigen-antibody reactions. Okay, so ELISA is mainly based on the interactions or the reactions between the antigen and the antibody. So infection by a pathogens, it can be detected by the presence of antigens, which is mainly a protein or the glycoprotein, or it can be detected by the antibody synthesized against the pathogen, because whenever any pathogen enters our body will develop the specific antibodies. So here in ELISA is mainly based on the interactions or the reactions of antigen and the antibody. So whenever an individual is infected by a pathogen, it can be detected by the presence of antigens, or even we can identify the specific antibody which is produced against the specific antigens. So here also we can detect the presence of the antigen at the initial stage of infection before the symptoms of the disease is visible in an individual organism. Clear? So this much for today's class. In the next class we will learn the further applications of biotechnology in the field of medicine. Clear? So this much for today. See you in next class. Thank you.